We are truly grateful for your presence and guidance. Thank you very much. We now move on an important segment of this inaugural session, that is presidential address. Before that, I would like to call upon Professor D.P. Sawan to introduce today's chairman of the function, Professor Arun Karatsar, who is going to deliver his presidential address before the keynote address. Over to Dr. D.P. Sawan.
Good morning, respected chief guests, distinguished speakers, all participants joining us from different parts of the country, and dear students. A very warm good morning to everybody present on this virtual platform. It is my immense pleasure and privilege to introduce the chairperson of this online session, Professor Arun Kharat sir, an eminent microbiologist, distinguished academic leader, and internationally recognized researcher. Professor Kharat sir completed his MSc in microbiology from Shivaji University, Kolhapur, and earned his PhD in science from prestigious Indian Institute of Science. He further expanded his global research exposure through postdoctoral fellowship at globally renowned Rockefeller University. With more than 26 years of teaching and research experience, Professor Kharat sir is currently serving as a professor at School of Life Sciences, Jawaharlal Nehru University since September 2017. Professor Kharat sir research areas are highly significant in present global scientific context. His specialization includes bacterial antibiotic resistance genes, post-pathogen interactions, environmental microbiology, antimicrobial and anti-cancer potential of medicinal plants. He has been honored with several prestigious fellowships and recognition. In addition to his research excellence, Professor Kharat sir held important academic leadership positions at different places. He continues to inspire young researchers and scholars across the country. On behalf of organizing committee, I extend heartfelt welcome to you, sir, and thank you for kindly consenting to chair this online technical session. Dear participants, let us welcome Professor Arun Kharat sir. Thank you.
Thank you, sir. It is my now privilege to invite the president of today's function, Professor Arun Kharat, a distinguished academician and visionary leader, to deliver the presidential address and share valuable insights with all of us. Professor Arun Kharat.
At the outset, let me morning to all the participant dignitaries on the dais and of the dais, Professor Dahabe, my very good friend and a colleague, even Professor Rathod, who is also my friend, and we have supervised a student commonly in the field of immunology. And Dharashi being my own district and the college where I have physically been there for a couple of times, it's really an honor to be a chairperson for a national level conference, which is being held on a very important topic. And it's very relevant also, especially nutritional security. I would take this at the center of the presidential talk. The time has evolved through the great revolutions, white revolution, Dugdha Kranti, and revolution, that's Harita Kranti. The green revolution certainly has given a birth to a whole lot of breedings, the varieties made crop possible, the goods are made possible, and people are able to consume the food. But at the present time point, it has come to the notice that what we eat, is it the nutritional value that we are consuming? And that is where the whole thing is rotating around. You would find that just keep aside the COVID as a pandemic, there has been growing concern around the globe, especially in the India, about the metabolic disorders, several different comorbidities, be it coronavascular disorders, be it a chronic or acute lung dysfunction, be it irritating bowel syndrome, irritating bowel disease, be it some fatty liver issue, which is a growing concern of a cancer these days, or the liver cirrhosis, pulmonary fibrosis, and the kidney dysfunctions. I'm not even talking about a diabetes. I'm not talking about a specifically identified certain conditions. The prostate, the breast, and all other cancers are also roaring largely because of deficiency of certain elements. And although it is very ironic, it is very strange that people keep on telling those who are sitting here of my age or around my age, their parents, their grandparents have told we have had a lot of crisis. Once in a 2 days, we hardly got the food. That is what people used to tell. And now overeating has become one of the concerns. If I may say, in the name of good looking, in the name of zero figure, anorexia is again an eating disorder. While consuming over the bulimia is another eating disorder. So no matter whether you are eating less, whether eating more, the question is that you are eating that you are supposed to. And the quantity versus the quality is becoming a very, very imbalanced. Another important issue, there is a people lost to the biodiversity. Very interesting, like people talk about beneficial plants, different beneficial aspects of the medicinal benefits of certain things. But then we are not increasing the content, we are rather ripping it off, we are rather taking it out and using it, consuming it without even replenishing even fraction of it. So this has posed a lot of difficulty in the in the nature, and also the global warming, the change in the tectonic plates and all other things is increasing temperatures, is also affecting the natural habitat of the plants, of the animals, and this is becoming many different species are on the verge of extinction. So because of that, again, becoming a limitation, and that becomes invites the technologist to come up with the solutions that would utilize the harvesting potential of those all plants, of those animals, but without affecting the biodiversity as a whole in nature. And therefore, it becomes very important for us to consider the environment, the ecology, the conservation of all biological forms, which cannot be considered without physical and chemical aspects out there. They are all interdependent. The quality of the soil decides the kind of metabolized chemicals presence and kind of chemicals and quality of soil. Physical properties of soil will decide the ability of the cropping pattern and the quality outcome, the quantitative and qualitative outcome of the crops. So eventually, whether we talk about jungle, whether we talk about a farm, whether we talk about a grazing animals in the farm and in the open land, everything comes back to the open environment, including human being. I often say, who has a control on the air? Nobody has a control on the air. I can have a 5-star like house, but I can't have a control on the air. I may have a air which is AC controlled within the house, but the moment I go out, I am not having any kind of control on it. And we have exemplified during COVID many deaths around the globe have associated with the well-to-do countries, developed nations, rather than the population. You would agree, or let's agree to disagree also. A lot of poor countries, including India, did not account the considerable mortality as it was expected. However, the developed nations have accounted considerable deaths during COVID. What I'm bringing the point out here is another. If I look into the India, within the India, urbanized cities and urbanized states, these are the two different things. Maharashtra is one of the urbanized states. And let's say Pune, Mumbai is urbanized cities of Maharashtra. So urbanized cities, urbanized state, and the economically sound population has suffered deaths or critical illness or intensive care units visit during COVID. What does it tell? It tells one thing for sure, that poor people did not have access, that one can always argue. But at the same time, who is on the road? We haven't accounted significant number of deaths on the road because of crisis. But we know there were deaths. There were high amount of deaths, and most of the deaths occurred in the good families, starting anywhere in the early 30s or late 20s, going up to the 60 or under 60 age. And this is probably the peculiar part of India, which was not the case in the developed countries. In the developed countries, people above 65-year age, they succumb to the death. And because ACE2 receptor was meant not to be very much in a quantity in the children, of course, the under 5 children who are having relatively poor immunity did not succumb to the death, did not succumb because they were not the etiological, they were not the targeted group for the pathogen. Interestingly, senior adults in the developed country and real adults, early young adults and the senior adults, all of them in India died, especially coming from the good family. I mean, those who are having provisions, those who are earning well, and they are having, but knowingly, unknowing, most of them have had a comorbid. You would see that. I always say it in my class that कोविड ने शुरू किया था, मगर अंजाम कोविड ने नहीं दिया था, अंजाम बैक्टीरिया ने दिया था, अंजाम पंजा ने दिया था. So COVID was a trigger to reduce the immunity quality, to prevent the moment it got compromised. Either the opportunistic pathogen from the nasopharynx, oropharynx penetrated deep into the lungs and increased the HRCT level, and thereby multiple systemic failure due to septicemia. Some of them, even without noticing, they may have died due to meningitis. At least lung failure and kidney failure and a liver dysfunction were the major causes, and largely they were due to the hospital community acquired pathogens. So definitely, if this is auxiliary infections, those are on topping up over the COVID was hospital community acquired infection, if not opportunistic infection from the own body. So you have to understand now, in the name of development, we have large number of population increasing to carry one or multiple comorbidities within, and they are the more vulnerable for heart attack, for arrest, for liver problem, for kidney problem, for gallbladder problem. These days, the removal of gallbladder is increasing. And I'm sure everybody must have heard at least one case in your known neighborhood, your known community, somebody has undergone removal of gallbladder. It's not a big deal. It's not a very surgery. Nowadays, it is done with a laparoscopy. But at the same time, it is interesting that the thing that we didn't hear 3 decades before, the thing that we didn't hear before 2000, we are hearing quite common, quite regularly. All these again boil down to the quality of your immunity, and quality of immunity is nothing to deal with your nutritional security. घूमा फिरा के बात वहीं पर आ जाती है. What I am eating, where I am living, do I get what I am supposed to get? Even if you take a Chavanprash, even if you take like Falana Dimkana, are these elements actually have the same nutritional value they are supposed to have in the books of Ayurveda? The question is that who has validated, who has verified, has done that detailed studies, and once you do detailed study, it will be an eye opener to figure it out. Actually, we are consuming them, but they don't have all those beneficial effects, mainly because the nutrient quantity has declined significantly. Some of the important element may be missing into it. That again, because the kind of water, kind of soil, these are non-biological. They definitely influence the biochemical reactions within the system, within the soil, within the plant, within the rhizobia, within the root, within the fruit, and those after consuming within our body. What in new 21st century, in last 15 years, one term has come up saying that One Health approach. And what is One Health approach? You cannot study animal as animal. You cannot study human as a human. That is beyond integrative medication, beyond integrative medicine. It is a health approach. Consider that all open ecosystem, environmental system, and we all living beings, be it microscopic or macroscopic, visible, complex, highly water associated, then zameen pe rehne wale, all those forms to be considered as a one Vasundhara, Vasudhaiva Kutumbakam, sort of that. And they are interrelated with each other. There is a talk ongoing, there is an exchange ongoing. We have to figure it out. And you will find that the root of many things will find in the water that we consume, many things that we'll find into the air that we are inhaling, and both of those we don't have. People have a good thing like salo setup kar lo, RO lagao, falana lagao. And now in last 10 years, people are talking RO water is not good for the health because you can't get rid of all divalent cations. They are required in the amount of parts per million. There is no rule of zero, no rule in the biology. Everything is a probability between the 0 and 1. There is never 0, there is never 1. So you can't get rid of all of the TDS and then say that I'm free of it. Then you have to add separately the minerals. In the name of those additional fortification of the water, we don't know exactly what is being provided, what are the sources of those material. And it is interesting that in India, you can get 1 liter water costing 8 rupees, and at the same time, you can get 1 liter water costing over 12,000 rupees. So it's kind of a strange. We have such a vast diversity, not only in the population, not only economic strata, not only the terms of caste verification, but we have a paying caste varies, and according to that, you are getting free water is available, 8 to 10 rupees per liter water is available, 10,000 rupees water available. Where are we taking it from? And then what is it doing in the body? The it enters in the body that is solvent to conduct all sorts of reactions. And interestingly, all those reactions are going to define the health of the individual, whether it is animal, cow, the buffalo, any other, and human. So therefore, here is a word of caution that now endeavor more research on a nutritional security so that we can bring the essential nutrient in the stoichiometric ratio so that the health of human being, health of animal, health of plant, health of microbe is maintained to the best possible. Therefore, we can ensure that all biochemical reactions would happen to the normality. So we will benefit, ourselves will benefit from all the harvest that we get it from this nature, from this ecosystem. And I hope, I wish all the very best to the organizer, to the participant for this next 2 days deliberation. And with this, I close my words here. Thank you so very much.
Thank you, sir. In your talk, you expressed your concern regarding metabolic disorder, cancer, disease, kidney dysfunction, post-COVID issues, and so on. Once again, thank you, sir, for your inspiring and insightful presidential address. Your valuable thoughts and guidance have greatly enriched this inaugural session and will certainly motivate all participants throughout this conference. Thank you very much. Now I request Professor I.S. Yanda to introduce today's keynote speaker, Professor G.M. Vidyasagar.
Hello. Hello.
Yes.
Respected chairperson, distinguished speakers, and respected participants and dear students. It is an great honor and privilege for me to introduce our keynote speaker for today's session, Dr. G.M. Vidyasagar sir, a renowned academician, researcher, and eminent scientist in the field of botanical science. Dr. Vidyasagar sir is currently serving as a senior professor of botany in the Department of Postgraduate Studies and Research in Botany at Gulbarga University, Kalburgi, Karnataka. He has an outstanding academic career with over 29 years of teaching experience and more than 32 years of research experience, making significant contributions to higher education and scientific research. His research contributions are remarkable. His research primarily focuses on bioactive molecules from indigenous medicinal plants, fungal endophytes, and soil microbes. His work has led to the discovery of antifungal, antibiotic, anti-cancer, and anti-diabetic biomolecules and therapeutic nanomaterials. On his major research achievements include documentation of more than 100 indigenous medicinal plants from the Karnataka region, highlighting their therapeutic applications and identifying 30 promising bioactive compounds. Professor Vidyasagar sir is recipient of prestigious uh, Commonwealth Fellowship and Fellow of Indian Botanical Society and National Environment Science Academy. He also served as a editorial board of the CEMU journal Science Philippa and is a life member of several professional scientific communities. Now, with great pleasure, I respect and invite Dr. G.M. Vidyasagar sir to deliver the keynote address. Sir, we welcome you and request you to kindly begin your keynote lecture. Thank you, sir.
Thank you, sir. Now everyone is waiting for the exciting keynote address. Now I kindly request Professor Vidyasagar ji to proceed for his keynote address. Hello.
हॅलो हा सर
सर नाही आहेत का ऑनलाईन?
आहेत जॉईन होतील
हो.
हो.
Your call has been forwarded to voice.
Hello.
Hello.
Can you hear me?
Hello.
आहे का क्लिअर?
सिद्धू येऊन जा.
हॅलो.
हा सर.
आहे क्लिअर?
हो सर, क्लिअर आहे.
स्लाइड दिसायला आहेत ना?
हो, दिसत आहेत.
ओके.
ओके.
So, can I start my presentation?
Yes, okay, sir.
So, thank you very much, the chairperson, all the delegates from different parts of the country, and the organizers. So, my presentation is on emerging therapeutic roles of plant bioactive molecules in multi-organ protections and metabolic regulations in type 2 diabetics. You all know the whole world is worried about different disorders, particularly the diabetic. Just a minute. Particularly the diabetic, and India in particular is largely struggling to manage the diabetic disorders in population. Is it clear?
Yes, sir.
Okay, fine. So therefore, I have chosen this topic since I am since last 10 years working on bioactive molecules from plants and microbes to manage the diabetic disorders. So I thought of sharing some of views with you all. Just a minute. Why there is problem with this key? There is some problem with slide sharing.
इनामदार सरला बोलो. इनामदार सरला बोलो. Slides not moving, sir.
Yes, slides are not moving. That's what.
Okay, one minute, sir. 1 minute. इनामदार सर, slide move होत नाही काय?
Is there any wrong with you or here?
सर F5 प्रेस करा. सर विद्यासागर सर, हॅलो. F5 त्यांचे
आवाज का येणं झालं?
हॅलो. माईक ऑन आहे का?
Yes, sir. Yes.
इथं आहे. ओके. थोडं स्ट्रक झालं. राईट. बरेपा.
ते म्यूट करून बोलायला त्यामुळे आता परत हे बघा म्यूट आहे, आवाज येणार म्यूट करायला सांगा. मित्र मेसेज टाकावं लागेल.
सर अनम्यूट आहात सर. सर म्यूट. अनम्यूट करा सर. अनम्यूट.
एक आलं की एक चाललंय बघा. आता आला. ओके. पण स्क्रीन हे होईना गेलं हो. एनलार्ज होईना गेलं. ते केलं तरी होईना गेलंय. ठीक आहे. इतका चालू का? हा. ओके फाईन. ಕ್ಯಾಮರಾ ಎಲ್ಲಿದೆ?
ಇಲ್ಲಿದೆ ಸರ್. ಸ್ಕ್ರೀನ್ ಶೇರ್ ಎಲ್ಲಿ?
ಇದು ಸ್ಕ್ರೀನ್ ಶೇರ್ ಕಾಣ್ತಾ ಇದೆ ಅವರಿಗೆ.
ಕಾಣಿಸ್ತಾ ಇದೆ. ಹಾಗೆ ಸರ್ ಸ್ಕ್ರೀನ್ ಶೇರ್.
ಓಕೆ. ಓಕೆ ಸಾರಿ. ಅದು ಮುಂದೆ. ಓಕೆ. ಸೊ sorry for the disturbances. So diabetes, type 2 diabetes is not merely the hyperglycemia. It is not only the increase of sugar, but it is also a systemic organal disease or inflammatory disorder we call. So it is a complex, very complex disorder. Involves several body organs, and finally it results into the increase in the sugar in the blood. So it is metabolic, inflammatory, mitochondrial, and also it is influenced with epigenetics or genetics also is very much involved, we can say. And all these parameters are influenced with the lifestyle disorder. I mean our lifestyle and lifestyle driven disease we can say. In the world today, we have around 537 plus million adults worldwide. And it is rapidly growing in Asia, and in that, in Asia, India again the very predominant country where diabetic is growing. So rising early onset, and we are witnessing with several symptoms. Nodi, stuck झालं बघा. No, there's something wrong in my system.
Sir, you can continue without slides, if you don't mind.
Without like, with
Without
mic
showing
only.
the slides. Oh,
Yes,
without
sir,
showing
yes,
the
sir.
slides, you can continue.
I think that is better, it seems so.
Yes, sir. We try to understand.
Okay, I think, is it okay?
Yes, sir.
Yes, sir. Yes, sir.
Okay, then so I will try to talk without mic or without slides.
Yes.
Okay, fine. Okay, so as it is genetics is involved, so there are several genes involved in this onset of the disease. So particularly one transcription factor gene, which is known as PDX1 gene, is involved very much. That is a transcription factor, and if this gene get activated, it binds with all the genes which are associated with the metabolism, glucose metabolism, wherever they are, in whichever the organelle they are. Most of the genes are attached with this one, and they get activated, and the PDX1 gene promote or enhance the production of several or activation of several genes which are related to metabolism. And if this particular factor is not activated, then there will be problem in the metabolism of sugar. So in this gene, there are 3 exons. Exons, introns, you must be knowing. See, this audience is PG and all other faculty members. It is. Hello.
Yes,
sir.
sir.
Audience includes PG students.
Yes, sir. Yes, sir.
And faculty.
All faculty members and PG students.
Okay.
And undergraduate students are listening.
So one gene which is responsible for transcription factor, that is known as PDX1, is very essential in the appearance of diabetic 2 or type 2 diabetic. So in this gene, there are 3 exons, and in that, 3rd exon is very, very important one, which encodes the hemodomain. It has 60 amino acids, which binds with the particular sugar molecules and activate. And if that is not synthesized, then there will be the diabetic symptoms in the person. It is transcription factor regulates insulin, glycokinase, and glucose T2 and somatostatin genes, and bind with the promoter, particularly in pancreatic genes, and control the pancreatic developments and insulin gene expression. So we understood the metabolism, glucose metabolism. So I have divided the lecture in 2 groups. One is the diabetic, the genetics, little bit genetics, not completely. So little bit genetics and the development of diabetic, and for next one, the bioactive molecules from different plant molecules. In the initial stage, let us understand how the diabetic develops. So since this one important gene is involved, its regulation is important. If it regulates properly, then it binds with wherever the promoter essential attached with the essential gene, so that promoter regulate the required genes. If it is not attaching with the genes involved in the metabolism, then sugar, the enzymes will not be synthesized and glucose will not be metabolized. So this is a key factor we can understand. So in the metabolism, now particularly we can divide 2 types. We have glucose utilization in two sectors. In one sector, we have intestine, liver, RBC, brain, neurons, kidney, and eye. So in these sectors, glucose is required, but no involvement of insulin in this. Insulin is not required to uptake glucose in these cells. Whereas in muscle and fat cells, insulin is very, very important. So without the involvement of insulin, glucose is utilized in these areas where with the involvement of insulin, the glucose is metabolized or used in muscle cells or fat cells or other cells. So now our important concept, both the things are equally important. Insulin is important in one case, whereas in other case, if insulin is not important, upregulation of glucose is most important here. I mean, that is damaging. Suppose in the glucose, if glucose intake is normal or glucose production is normal, then there won't be any problem. If slight increase in the glucose production occurs in the body, then automatically in all the cells, brain cells or liver cells or RBCs or sometimes intestine cells, kidney cells, without any support, the glucose will be entering into the cell and all the essential activities will be normal. Whereas in case of tissue cells, so there is need of where there is need of insulin, then insulin work like the transporter here. Insulin work like transporter, and some substrate molecules in the cell, they act as the opener, door opener for the glucose. So together insulin activate the door openers, that is the substrate molecules, and there are several enzymes involved in this, and those enzymes get activated and open the door for glucose in presence of insulin, and then glucose enters into the cell. And once it is entered into the cell, particularly in fat cells, then the metabolism starts, and we know very well how the glucose is metabolized with glycolysis, then mitochondrial activities, and finally the electron transport system. So all these, of course, the glycolysis occurs outside the mitochondria, and in mitochondria, when it is two molecule carbon molecules enters into the mitochondria, then we know the respiration and the all molecules, energy rich molecules, whether it is in to these molecules pass through the electron transport system, and finally ATP generates, and ATP used for different mechanisms in the cell. This occurs in the tissue systems. Tissue systems are fat cells, whereas in other, direct glucose is entering into the cell and it is one difference is there. So mitochondria, though in some cases are there, so only the glycolysis operates in the brain cells or even in the cells, and whatever the glucose released out of it, so that is used in the activities in that particular area. Even though mitochondria is present in some cases, mitochondrial metabolism is not occurring in these cells. Okay, so glucose now is utilized in two ways, one with insulin or one without insulin. Then, so in kidney, glucose enter in largely, it is known as insulin dependent. The mechanism is slightly different, as I told. So there are two ways in the kidney. One, it filters the blood and again removes the this one goes and again absorb the glucose and retain into the blood. Sometimes it the sugar molecules released and some sugar is coming out and some sugar again go back to the blood. Blood sugar again retains. So most glucose handling occurs in the proximal areas, and here particularly the glucose T2 and T1 transporters are important in kidney. As in mother cells, T4 transporter is essential. Then let us see the other one is glucose and glycogen conversion. Whenever glucose concentration increase in the body, so as we know, so it get converted, most of the glucose, if it is not used, it get converted into glycogen in the liver and as a deposit in the liver. And whenever it is required, again the glycogen get converted into glucose and transferred to the cells or different body cells, and it is used. This is the mechanism that we have already understood. Now coming to the core pathophysiology. Core pathophysiology of diabetic, type 2 diabetic is insulin resistance. First is insulin. One is the progressive beta cell dysfunctions, and third one is chronic low grade inflammation. Next one is the mitochondrial dysfunction, and lastly, so these 5 symptoms we can see when there is type 2 diabetic. First is insulin resistance. It appears when there is more glucose in the initial phase. If the glucose concentration is more in the body, so beta cells start insulin. And even if the increased insulin level is not able to send the glucose into the cell, then automatically the insulin production get reduced. And we call that it is the resistance, insulin resistance. Then we have another one is the progressive beta cell functions. So beta cell function also is getting affected because of the more pressure in the production of insulin, and in the later there will be dysfunction in the beta cells. Then slowly we get chronic low grade pain in the body throughout the day or throughout the night. Always pain will be there in the body, and this is also another symptom. So it is because of other things I will tell you in the later stage. Then mitochondrial dysfunction. Mitochondrial dysfunction, because most of our metabolism occurs in the mitochondria, and all the involved. Interesting feature here, mitochondrial feature is mitochondrial genome is different from the human other genome, nuclear genome, which is exposed to the free radicals in mitochondria. Generally, if the sugar increases, then there will be more free radical generations in the mitochondria. And when there is more free radicals in the mitochondria, then there are chances of damaging the cell membrane and even DNA. Why DNA? Because DNA in the mitochondria is exposed to the free radicals. In other cells, in nuclear cells, the DNA is coated with the histone and nucleosome is formed. It is well protected by the histone. Whereas in mitochondria, histone is not there and DNA is not protected well. So therefore, the free radical generated directly affect the DNA sequence, make the damages in the DNA level. Many a time in the later stage, mutations are possible. Okay. Now, as a result, what we see here is in the protein, there is a substrate known as insulin, which is protein. So, where there are two proteins, serine, another one is tyrosine. In the normal stage, the always insulin resistance in condition in which responds poorly to the insulin signaling. And as a result, the pancreas produces more insulin and serine of the protein, which is phosphorylated instead of tyrosine. In the normal case, tyrosine is always phosphorylated and leading to, in abnormal cases like diabetic 2, if it is serious one, then in place of tyrosine, serine will be phosphorylated. And you will find if you test the blood, so you will find more amount of tyrosine in the blood. That is the indication, or it is the marker we can call if there is diabetic. Directly, if you just test the presence of concentration of tyrosine in the blood, so that indicates that the stage is diabetic or not. Then if a serine is phosphorylated, then it indicates the diabetic stage is achieved. Elevated plasma tyrosine is a biomarker for insulin resistance, and serine deficiency linked to diabetic neuropathy and poor wound healing. So we have one more project with the UK, and we are working over this on particularly the wound healing mechanisms, and we are planning to nanomaterials and biomolecules. So a composite we are making, and that composite is being used in the treatment of skin ulcer in diabetic patients. So serine supplementation sometimes are given in the form of drug, which inhibits the production and accumulation of toxic lipids. So it is a management this one therapy. So progressive beta cell dysfunctions, the pancreatic beta cell producing more insulin to maintain blood growth. So in chronic high glucose and high fatty acids, then the mitochondrial overload, particularly with ROS productions, and also the stress, ER stress also is seen, and inflammation starts. Then stress kinases enzymes also start producing, and these leads impairing in insulin secretion and in first phase insulin response. Insulin response is lost when there is ROS production in the ER and mitochondria. Then beta cell apoptosis also possible in the later stage. So later stage, if care is not taken, then there is a possibility of beta cell apoptosis and reducing number of beta cells, and that stage is, I think, no reversible. In very rare cases, there will be this one stage. So beta cell produce less and insulin level falls, and then the result diabetic seen. In chronic low-grade inflammations, what happened is, so the pain is very slow, but it is continuous, and it is metabolically driven, we call, where there are cytokines. There are several different types of cytokines which are produced, and it is metabolic related only. And these cytokines are continuously generating or producing in the body, and low-grade pain continuously seen in the body. Excess nutrition that leads to adipose inflammation, then cytokine productions, and finally it results into insulin resistance and inflammation seen, and inflammation not taken care of in the early phase, then it leads to obesity. And finally, insulin resistance, beta cell dysfunctions, and finally, vascular complications in the patients. See, again, the mitochondrial dysfunctions we have seen, and metabolic inflexibility is also seen in the patients, where the lipid and glucose oxidation, lipid to glucose and glucose to next oxidation to insulin resistance. Sometimes to switch between lipid and glucose oxidation in response to nutritional state, so contributing to insulin resistance in many cases, along with the other complications. We have some discussion which is and which the mechanism is gluconeogenesis. So from glycogen, gluconeogenesis is the main function, and also it protects some other organs as well. There are other medicines also, which are commonly and it also is useful in AMP pathway. AMPK pathway is activated through this metformin, and beta cell protection also is by this particular medicine. Therefore, we generally consider that metformin is safest market. Now, I think this is overall background we have seen how the glucose, if it increase in the blood, how it creates complications and damage the cells, damage the DNA, and results in the form of type 2 diabetic. Now, on the other hand, we have several medicines. Several we have seen Ayurveda and many other traditional systems reveals that there are a number of plants present in our country, and they are in use for the treatment of diabetic. And of course, the properties of these medicinal plant varies with region, varies with conditions, varies habits, even food habits also, our genetic makeup as well. So some of the plants on which we have worked and we have been working on this is, one is the Gymnema sylvester. Gymnema sylvester, where the active compound is gymnemic acid, and its property, it reduces intestinal glucose. So even in the intestine also, as we have seen, glucose is absorbed without involving the insulin, and regenerate beta cells. So the property of this gymnemic acid is reduce the absorption of glucose in the intestine and regenerate beta cells. So it is reported in the experimental model. Next one is the Momordica charencia. The active compound is cherencin. Then polypeptides we have there, and some compounds like insulin have seen, and they are also involved in the management, are involved in the reversal of diabetes. Then we have curcuma longa. Curcuma longa, the active compound, which is very, very important and common, used in majority of disorders, which is known as curcumin, which reduce insulin resistance and anti-inflammatory actions. Then pterocarpus we have, it contains epicatechin, and epicatechin is very important in the protection of beta cells, beta cell damaging. Then the property of all these bioactive molecules, multiple properties we have. See, it enhances insulin secretion. It stimulates pancreatic beta cells, protect beta cells from oxidative stress. See, oxidative stress is the most or primary important we can call in the diabetic patients. And once there is oxidative stress increase in the cell, then there will be damages of all the organelles or the components of the organelles, and function of metabolism get disturbed. So therefore, we have to first manage the oxidative stress and improve incretin response. And alkaloids, flavonoids, saponides, majority materials, compounds are involved in this. Then there are some improving insulin sensitivity. This is also important. One is secretion of insulin, and another one is sensitivity also. Sensitivity to bind with the other substrate molecules. So that also is equally important. And we have molecules, and these molecules, even the modern drug metformin is also doing the same work. Then inhibition of carbohydrate digestion. So carbohydrate digestion is also another mechanism which we need to control the diabetic. So here these drugs involved in the either reducing the digestion, carbohydrate digestion, or slowing down, we can call, particularly the enzymes activity like alpha amylase inhibition or alpha glucosidase inhibition, or sometimes delayed absorption of glucose in the intestine. Then antioxidants and anti-inflammations, as I told in the beginning, both are equally important. So therefore, reducing the ROS generation is the prime duty, prime responsibility of every drug. So the herbal drugs are involved in such activities. Then lipid profile modulation is also another important activity. So plant drugs are taking care of these property. So primary target is multi-organ because liver, muscle, and adipose tissues are involved. So in this case, insulin is involved, and the primary metformin is working here. Then plant drugs are also simultaneously working. The advantage of plant drugs. So in plant drug, we are giving acting against what? What is the activities? It is different, and the activity in good. So therefore, many people trying to work on, but it is not possible to identify a specific activity of specific compound. You may get specific activity, but when it combines with other molecule, its activity will be different. So therefore, herbal drugs, that is complex drugs, are always good. Without knowing one or other way, they are supporting and they are useful in overall complex disease management, like diabetics. So multi-target action is there. Then side effects are very low because all these drugs are natural, and in our body, our gene or our defense mechanism know well that how to metabolize natural compounds. So since they are natural compounds, our defense mechanism metabolize and without any residual effect, body manage the metabolisms. Whereas in the modern drugs, sometimes metabolism will be delayed, or sometimes there will be the drugs and accumulation in different sites in the cells, and those accumulated drugs later will have side effects from different mechanisms. So therefore, the natural drugs are useful in this way. Then they are suitable for long terms. The plant drugs are suitable for long terms since we use in small dose. So side effects will be very less, but without knowing the dose, because in plant particularly, as I told, during different season, different molecules will be synthesizing. Different time of hours, the different molecules will be synthesized. And the molecule which is involved in the diabetic management, we don't know in what time the particular drug synthesis of plant. And unfortunately, if it is when we are collecting, and that time if the drug concentration is more and we don't know, and without knowing, if we consume leaf instead of one leaf, if we take 2 leaves, then the drug concentration will be very high, and there are possibilities of sudden damaging the tissues or very severe effect seen in the patients who are taking herbal drugs without any doctor's suggestions. So therefore, it is not advisable to take herbal drugs without any doctor's suggestions or prescriptions. Then, of course, the traditional systems have proved, but those traditional healers are experts. They know how much to be given, and if it is found to be toxic, then what is the other measures to reduce the toxicity. So they are the known people. We should take their advice, or some practitioners, registered practitioners, and take the herbal drugs. Okay, now we have some polyphenols which have got antioxidants and anti-inflammatory properties along with anti-diabetic properties. Curcuma longa, for example. And from grapes, some polyphenols identified. One of these is the resveratrol. So this has got antioxidants properties, cardioprotective properties, and also the anti-acid. Even the onions or apples also have shown some flavonoids with anti-inflammatory, antioxidants, and anti-diabetic properties. Even in soybean also, isoflavin has been isolated and shown the properties of antioxidants and anti-diabetic. Catechin green tea is largely now recommended in the diabetic case, where it has got antioxidant, anti-cancer, and it is one they have got some anthocyanins are important in the production of and the protection of the organs, different organs. And so all these properties, different properties like antioxidant active properties, anti-inflammatory properties, cardioprotective properties. Hello. So all these properties are there in bioactive molecules extracted from the plant. So with these properties, if any drug is available, one or two drugs together with all these properties, then probably it is very, very useful to manage the symptoms of diabetics, or also we can achieve the of the diabetic with this one. So simple understanding of this. So plant bioactive molecules, they have antioxidants, anti-inflammatory effects, they improve beta cell functions, and they activate like PDX1. So PDX transcription factor gene is being activated, seen, and that results in increased synthesis the plant metabolites. So therefore, these are normally used and in the treatment of. And in short, so I can conclude here. So in conclusion, some points say, so plant bioactive compounds have gained major attention in managing metabolic disorders such as mellitus. So compared with many modern synthetic drugs, plant bioactive molecules often provide multi-target therapeutic effects with fewer side effects. It is essential to think over this and start as far as research is concerned, I'm talking about. However, numerous plant-derived bioactive compounds exhibit anti-diabetic properties. Their direct molecular regulation of the PDX1 transcription factors remains poorly understood. This is very important. At a global level, the PDX1 transcription activation, we thought when we give the drug in complex, it's seen that this particular activity is seen or is there in particular compound, but lots of studies have to be conducted and the result should be, right now it is very poorly understood, but still work on this. And another one very important is the epigenetics. We always see that every mechanism in our body now in 21st century, in the last of 20-30 years, people have identified and now identified in several cases that every gene has got one or other way it is influenced with epigenetic mechanisms. It means biotic or abiotic factors within the body they all are one or other way related for the regulation of genes and resulting in either healthy conditions or diseased conditions. So epigenetic is also important involved here, and it has to be studied well with some examples like plant metabolites. Then very few studies have explored whether bioactive molecules modify the epigenetic regulations of these or not. Very few studies, still lots of scope is there to work over this. Most research on PDX1 regulating bioactive compound is limited to cell line animal models, only cell lines. Now, whatever we are studying, so all the studies either we do lines, cell lines, or with one or two other kind of animal models. But clinical evidences are very scarce, are very rare, we can say. So clinical evidences are required to release any drug in the market. We need to have large scale studies on human beings, and then only it is acceptable. So therefore, now finally what I conclude is plant drugs are the complex drugs. They have many properties, as I told, 4 or 5 different properties they have got, and all together are working and managing different types of diseases, complex diseases like diabetic. So diabetes is not only because of increase in the glucose, as I told, it is because of so many issues related to different organs. And finally, the diabetic results. So to treat disease like this, complex disease, we have to have a combination, and the herbal drugs are common. So these are the good source or best source to make the valid drugs in the management or treatment of herbal as compared to the modern drugs. Whereas in modern drugs, it is only one single compound. It has got one property. Every time one particular property is managed or treated. And in diabetics, since there are several issues to be resolved, so one drug may not work properly. So it has to be multiple drugs. It should be some multiple drugs together given for the patients to treat. So scientists at global level are working both in modern system and also in the herbal systems and coming out with different results, but still lots of, because the parameters are the causing factors are also varies with the different diabetic patients. All factors are not similar in every patient. There are different factors, and different factors with different disorders needs different kind of medicines. So therefore, still proper drug in the herbal area is not available or valid. It has to be validated. So this is what I wanted to tell you, how the stage of herbal drugs or how they are useful in developing the modern drug in the 21st century, particularly in like country where we have maximum plants, 7,500 plants. We have medicinal plants, and among them, there are number of plants we are using in the management of diabetics in different regions in the country. So all these plants we have to scientifically validate, and then only we have to say something about them that these are safe and can be taken up and used in management. So thank you very much. If you have questions or anything to ask, you can please discuss or raise a question. Thank you.
If is having query regarding this talk, they may ask at Vidyasagar. Okay. Thank you, sir, for your excellent keynote address.
I'm really sorry. I think there is some error here coming, and it is not visible. So I'm extremely sorry for my presentation.
No, sir. Despite of that issue, you spoke very well. Thank you very much.
Thank you.
So once again, for your excellent keynote address, your informative and inspiring presentation has truly broadened our perspective on the theme of the conference. By sticking with the theme of the conference, your talk, you spoke on role of plant bioactives in multi-organ protection and metabolic calculations in type 2 diabetes. Also, use of plant drugs in type 2 diabetes. We sincerely appreciate your time and valuable attention. Thank you once again.
Thank you very much.
Every successful event deserves a sincere note of gratitude. To formally conclude this inaugural session, I now invite Professor D.M. Pathan, Head Department of Zoology, Mahavidyalaya Gunjoti, to kindly deliver the vote of thanks.
Respected chief guest, distinguished speakers, distinguished participants, faculty members, and dear students, a very pleasant good afternoon to everyone. On behalf of the organizing committee, it is my proud privilege to propose the vote of thanks for this online inaugural function and keynote address of our conference. First, I express our honorable chief guest, Professor Arvind Dhabe sir, for sparing valuable time for a busy schedule to inaugurate this conference and enlightening with us inspiring words. Your guidance and throughout flowing address have truly set the tone for the session ahead. I extend my sincere thanks, Chairperson Professor Arun Karat sir, School of Life Science, JNU University, New Delhi. Your presence has added great significance to this event. My special thanks, Professor GM Vidyasagar sir, Professor and Head Department of Botany, Gulbarga University, Kalburgi. He has delivered keynote address for sharing this knowledge, experience, valuable insights. Your presence has added great significance to this event. I would like to thanks to all technical team members for coordination throughout the session. A big thank to all the participants, research scholars, faculty members, and students who have joined us today from different places. Your enthusiastic participation in the true strength of this conference. Lastly, I expressed our gratitude to our sponsor. Lastly, with this, I concluded vote of thanks. Wish the conference great success in the upcoming sessions. Thank you to one and all. Now this program is over. After 10 minutes to start the technical session, first in Bharat 10 to 1 p.m. Over to Vijayapuri sir.
Thank you, sir. As you said, the first technical session will resume after 15 minutes break. Thank you very much.
कमी सर आता
त्यानंतर बघा की गुड आफ्टरनून चंद्रशेखर सर.
Good afternoon, sir. Good afternoon.
आफ्टरनून.
Good afternoon, all of you.
गुड आफ्टरनून डोले.
Good afternoon, sir. Good afternoon.
बाहेर येतात का जैन जैन हा म्हणा कुठं आहात?
Good afternoon, Patan sir.
गुड आफ्टरनून. गुड आफ्टरनून पाटील सर.
सगळे हे दिसाले. माईक चालू आहे.
Can you start, Omerga sir? Dr. Omerga.
शुक्रवारचं
Omerga sir join झालं का? हॅलो.
मला पाठवतो वाटतं तिकडं नाही झालं आजची ऑनलाईन मग मी आपली बाहेर आले.
का जॉईन झालीत का? झालीत आहे. जॉईन झाला का? आवाज येणार झाला?
होतो लगेच.
वा वा, एकदम चालू करायचं. हा, आवाज येतो का माझा? बस, थँक्यू.
आणि ते आपलं पुढं कुठं तर त्यांनी ते त्याच्यात नंबर आलेला पुढं पाठवतो म्हणले.
स्टार्ट सर. चंदन शिवे सर, ढोले सर, आवाज येतो का?
यस सर.
यस
यस सर.
ओके.
वाघमारे सर स्क्रीन विजिबल होत नाही तुमची.
कुणाची? माझी?
वाघमारे सर स्क्रीन
आहे विजिबल आहे. शेअर नाही केले की मी करतो.
ओके. गुड आफ्टरनून टू
स्क्रीन शेअर
ऑल.
करा ना तुमची.
Good afternoon to all respected distinguished speakers, members, teachers, scholars, and dear students. For this technical session, first, I would like to invite the chairperson of this technical session, Dr. Chandan Shive sir, R.G. Shinde College, Paranda, and the co-chairman I have invited as the co-chairman of this session, Dr. Dole sir, SDM College, Kalam, and for this session, technical session, I would like to invite the speaker of this session, Dr. Somnath B. Wagmare, sir. It gives me great pleasure to welcome to all technical sessions of our conference on recent trends in biological sciences for sustainable development 2026. The technical session are the heart of the any academic conference as the provide platform for the sharing knowledge, innovative ideas, and recent research development. Now we have privilege to have with us eminent personality as our invited speaker for this session, Dr. Somnath Wagmare, sir. I have briefly introduced the resume of Wagmare, sir. Dr. Somare is associate professor, recently working as a associate professor in Department of Zoology in overseas Wadia College, Pune. Dr. Somnath Wagmare, sir, has awarded award, scientist awards, Professor Satya Yamaguchi Parasitic Taxonomy Promotion Award, Senior Scientist Award, Recognition Award, Senior Scientist Awards. He has also member of the Society Nematology Society of India, Indian Society of Parasitology, Zoological Society of India, International Association of Zoologists. He has published 51 papers in national international journals. He attended national international conferences and delivered lectures in different places in our country. He attended 32 workshops, conferences, so and so. He has research guides under his guidance. Currently, 3 students are working for the PhD registration. He has also recognized MPhil guides. His research specialization, the applied parasitology, nematology, systematics, biochemistry of cystodes, molecular modeling, structural biological of parasite proteins, molecular modeling, prediction of 3-dimensional structures of proteins and enzymes of helminth parasites, homology modeling, molecular docking. So this is a research area of the Somnath Wagmare, sir. So this is I have delivered or expressed the short resume of the invited speakers. So it is my honor to Dr. Wagmare, sir. Today, the topic of the invited lecture for sustainable development. I am sure this lecture will provide valuable insights and inspire us with new perspective in the field of biological sciences. Without taking much time, I humbly invite Dr. Somnath Wagmare, sir, to deliver the invited lecture under the chairperson of the Chandan Shive, sir, and co-chairperson Dole, sir. Over to the chairperson and co-chairman person of this session. Proceed, Chandan Shive, sir. Sir, proceed.
Sir.
Good afternoon, everyone. Shrikrishna Mahavidyalaya Gunjoti organized today National Conference on Recent Trends in Biological Sciences for Sustainable Development on today, 6 March and 7 March 2026. Department of Botany and Zoology organized this type of the conference. I'm happy to announce that today's chief guest, Dr. Somnath sir, is delivered very useful and important topics. So I request Dr. Sir, Somnath Wagmari sir, to deliver your valuable speech on such topics.
Thank you, sir. Thank you. I'm audible. Sir, I'm audible, sir?
Yes, sir.
Yes.
Okay.
Yes.
Okay. So, good afternoon to all the participants. And most importantly, the organizer of this two-day national conference on the recent trends in biological sciences for the sustainable development. Dr. Pathan, sir. Then chairperson and co-chair of the session, my friend Dr. Chandan Shive sir and Bhule sir. So I'm going to present the slide here. Just let me know. So my screen is visible?
Yes. Sir.
Okay. So the Patel sir, what is time for me? How much time is there for me?
10 to 1. 50 minutes.
Okay.
50 minutes.
Yes, sir. Okay. So without getting the time, we'll start to present my work in this session. So as the title of this conference is the recent trends in the biological sciences for sustainable development. So by considering the theme of this conference and as per the suggestion of the organizers, here I'm going to present my research work and try to correlate my research work with the theme of the conference, that is sustainable development. So the title of this my presentation is Nematodes for the Sustainable Development. As we know that the nematodes, they are very important one and very useful creature. And we also know that the nematodes, they are everywhere. The nematodes, if we try to say some of the basic structure of the nematodes, they are the unsegmented, we can say the non-segmented worms, and everywhere the distribution is there, and the distribution is in different habitat, and approximately every ecological extremities, like from the fresh water, marine water, plant, soil, everywhere you can find. These are the very least studied organism. Still, if you see the diversity, in my presentation, I'm going to see the important facts about these nematodes. So we'll get amazed that so much diverse habitat is there, and every habitat you can find this nematode. So these are the most critical equilibrium maintaining factors and having implications on the yield of both health perspective also, and the health of both, that is animal health and plant health. Both the concern, these nematodes, they are very important animal. And however, their taxonomic knowledge about this nematode is very scarce. So in my today's presentation, as per the suggestion from the organizers, I am going to present nematode as a sustainable development. And at the same time, most of the research workers, they are doing the research in the nematodes. And I'm going to highlight what is the recent trends in the nematology research, as well as what exactly I am doing here in nematology in my lab. So basically, this is just outline for the classification of nematodes. So here, as we know that the nematode, they are very diverse group. You can find nematode everywhere, which is in the course of evolution. You can see this is the fourth or fifth phyla. where you can find the nematodes. And if you see in the process of evolution, most of the body organs, they start to develop from this nematode. So in the evolutionary point also, these nematodes, they are very important. Yes, they crucially for the sustainable development. So nematode be broadly classified into the three groups: free living one, predatory nematode one, and the parasitic one. And again, if we see their parasitic nematodes, again they are the human parasitic, they are the vertebrate parasitic, they are also present into the invertebrate, and most importantly, they are also present into the plants. So in my presentation, I'm going to more emphasize on the plants because this is closely related to the sustainable development. And if you go through the sustainable development goals of the US, there is a 17 sustainable goals is there, and this nematode they are present in 4 sustainable goals. So what are that 4 sustainable goals by the US nations and how these nematodes are related with that, we'll also see. But in my presentation, I'm going to concentrate more on the human being and the plant nematodes. And if we're talking about the nematode and why the nematode, they are matter for the sustainable development, because the nematodes everywhere, we know that they are important for the soil health, because the nematode, they important in the nutritional cycling. We also used the nematode as a bioindicator of the soil, bioindicator of the water, particularly the pollutants. They are also important for the pest management, climate action, particularly carbon sequestration and soil diversity. At the same time, these are nematode important for the managing the waste material, the decomposition, then the natural pest control also. So overall, if we see the nematode, they're very much important, but they are the neglected group in the kingdom Animalia, and they're very much close to the sustainable goals. Here you can see the goal number 2, goal number 12, goal number 30, and goal number 15, which is the US nations sustainable goals, which is there in all the humankind. So you can correlate nematode with the zero hunger, as we know that because of the nematodes, there is a extreme loss of the health, that is both plant health and the crop health. Altogether, they are damaging. Then the consumption also, responsible consumptions. Then for the climate actions, then the life, so the nematode, yes. If you take the 100 cc of soil, you can see the thousands of nematode in that 100 cc of soil. So you can see the how much important this nematode is there. And more importantly, they are the hidden engineers of the ecosystem. You can also see in the context of the plant, you can see the hidden enemies. Okay. So altogether, nematodes, they plays a very important role and they are very much closely related with the sustainable development. So here, if we see the nematodes and their abundance on these all kingdom Animalia, so general characteristics, we know that the nematodes, there we see the body is very simple body, transparent body, and here there is a very important cuticle is there, and you see their habitat. You can find these nematode everywhere, into the freshwater bodies, into the marine water bodies, into the deep seas, into the plants, related to the animals, vertebrate and invertebrates. Okay, so and there are very some parasite, some nematode, they are the parasitic, some of they are the free living. Again, the parasitic, there is a different kinds of the host is required. So basically, these are the 2 nematodes which are very important. One is the C. elegans, and another one is the Melodogani incognita or Melodoganus. This is related to the plant one, and this is the free living one. But if we see as an animal model, in particularly our subject zoology, we use the different kinds of animal models, and the C. elegans is very much famous model because till date 4 Nobel Prizes were awarded. Okay, the different scientists they use this C. elegans as a model organism. There is all kind of nematodes. They are important for nutritional cycling, biological control. Okay, so this is the significance for the sustainability. So these are also important for the essential indicator of environmental health, and they are also important for the carbon cycling. So this is also again the sustainability. The role of nematode is very important one, as in our knowledge, and particularly whatever the syllabus we taught into the school and colleges, where we highlight nematode most of the time as the parasites. No doubt they are the parasitic one, and they are harmful Yes, but not all the nematode that are parasitic and not all the nematode that are harmful. There are some useful nematode is also there, non-parasitic nematode is there, free living nematode is there. So now there is a need to concentrate nematodes not only in the perspective of their parasitic nature. Now we have to concentrate this nematode with the other perspective also, and more importantly, how we can utilize the capacity of this nematode for the sustainable development. Okay, so that's why I'm congratulating the organizer for choosing a very nice theme, the sustainable development, because nowadays without sustainable development, it is not possible to survive for anyone. Now the next one, there are some interesting and very amazing facts about the nematodes. That's why, which is called as the ubiquitous roundworm. Why so? If you go through this characteristics or these facts, you come to know that they're about why these are the ubiquitous. The first one is their abundance. Every time we know that we say like that, nematodes, these are the very dominant in nature. That's why 4 out of 5 animal from the earth is nematode. So you can see that every 4 animal, out of 5 animal, 4 is a nematode. They found everywhere, the soil, ocean, plants, etc. Then their diversity, near about 30,000 described species is there from the 1 million estimated population of nematode. So we only come to know about only 30% of the population, and among that 30,000 described species, 6,000 these are from the these are related to the crops. Okay, then if we are talking about the losses of the nematode, so yearly near about $175 billion is direct loss because of the nematode. So for the sustainable development, the nematode control of the nematode is a very important one. As we know that they are useful also. Yes, they are useful as a model organism. So this is the first genome sequence of the C. elegans model organism, and the scientists, they got the 4 Nobel Prizes for studying this C. elegans. Okay, this is the C. elegans, which is having the very fully mapped genome. And why it is a model organism? There's very short lifespan period, 2 to 4 days to reproduce. They are also parasitic to the plant. Almost every plant, you take a perennial plant, you take annual plant. So herbs, shrubs, you take anyone. All the plant, they are having lots of infestation because of these plant parasitic nematode. Particularly, they do the damage by making the knots at the roots, and altogether the plant is unable to survive. And particularly the farmers, they are facing the problem of these nematodes, particularly the pomegranate growers, then soybean, in potato, in every plant you can find this problem of the nematodes. That's why the government of India, along with in the ICR, Indian Council of Agricultural Research institutes, along with the nematology, along with the entomology, the government is now highlighting about this nematological test. One side, these are important, they are very abundant, they are useful for the model as a model organism, they are very short time, their life cycle, they are parasitic also. But at the another side, they are also beneficial nematodes. We can use these nematodes, particularly the entomopathogenic nematodes. So these entomopathogenic nematodes, they are also particularly used for the biocontrol of the insect pest. So how at one side the nematodes are damaging the roots, and another side whatever the pest is there, again this pest comes to the attack towards the crop and they damage the crop. So we can use the same nematode which is damaging the root of the nematode. We can use the nematode present into the soil for controlling this pest. So this capacity of this nematode is known as the entomopathogenicity. So this nematode we can use to kill this insect pest. In this way, we can use the nematode for the biological control. As because of the excessive use of the pesticides, synthetic pesticides, now there is a resistance is also there into the insect. And because of that, there is a over spread of the pesticide, which leads to the development of the different type of cancer, that is skin cancer, lung cancer, intestinal cancer. So why? Because of the excessive use of these pesticides. So if we replace these pesticides with this biological control by taking the help of the nematode along with the fungus, along with the bacteria, then it will be it will really helpful for the nature, and in this way we can produce the crops, we can produce the healthy soil also. At the same time, the nematode which is present into the soil nematode, particularly the predatory nematode and the fungivorous nematodes, they are present into the soil and they important for the nutrient cycling, for the maintaining the soil health. This nematode, they are very important. So see, particularly into the agricultural sectors, when we spread the nematicides to control the nematodes, the nematodes that are present below the soil near about 10 to 15 centimeters below, and whatever the nematicides, pesticides which are spreading is above the ground. So basically, the problem is at the root, and we are spreading the nematicides on the above. So it will take lots of time to reach that particular nematodes. It's 10 to 15 centimeter, you can say that. So, so the thing is that to control the nematode, we are using the nematicides, and whatever the actual requirement of these nematicides which is needed and how much we are spreading. So a huge gap is there. So it causes the burden on the farmers also, and at the same time we are polluting the soil. At the same time we are polluting the soil, and at the same time we are killing this good nematode. These parasitic nematodes which is present into the soil and they are making the knots. We are not killing this parasitic nematode because already they are present into the plant roots cells and they are present into the free form. So whatever the these pesticides which are using, they first kill the nutritive recycling characteristics showing nematodes, and then they kill the root knot nematode. So at one side we are not succeed to kill the parasitic nematode, but on the other side we are killing the good nematode which is helpful for maintaining the soil health, which are important for the nutrient cycling, recycling. So this is very interesting fact which need to consider while doing the nematology research. And another thing is that they are very dominant also, and they are extreme survivor. They are extreme survivor. As we know that nematode, they are present everywhere from the soil is there, from the vertebrate, from the invertebrate, from the different animals, then free living form. At the same time, recently we got some records from the nematodes from the Arctic and Antarctic regions. These are extreme survivor. So they are surviving during the freezing and dying. So that extreme cold environment, these nematodes, they are able to survive. And there are some space expeditions. There's few reports of the space nematodes also. It means the nematodes, they are present into the space. And yes, these are the nematodes which is present everywhere. They are damaging like anything. That's why if all matter except the nematode disappeared, the Earth's outline would still be visible. So you can say that how much dominant is there for this nematode. And if we see the nematode, this is all the molecular evolution tree of the nematode. You can see these are the 1, 2, 3, 5 to 6 that order is there, and these are the nematodes. These are few representative nematodes. And in this, you can see some of the plant parasitic one, vertebrate parasite, invertebrate parasite, and the microbiome. So basically, this plant, you can see they come under the Tylentomorpha, the species Melodogyne, Incognita Melodogyne, Javanica, Hapless. So lots of species is there. So they come under the Tylenchilla. Then you can also see the Inoglea, where you can see the Zipinema, which is also known as the dagger nematode, okay, and the Dorilamida. So these are the most dominant nematode species which we find in our area. So these are the trophic complexity. Nematode occupy almost every functional roles in the food web. Like the bacterivorous nematodes you can find the fungivorous, you can find the predatory nematodes, you can find the plant parasitic nematodes. Okay, so basically in all the groups, the plant parasitic nematodes become overwhelmingly dominant. Sometimes about 50% of the community. These are the plant parasitic nematodes. If we're talking about the numbers, these are the most dominant, that is the plant parasitic nematodes, and they specific high intensity agriculture or specifically environmental gradients. So they are very dominant. Next point is that why to study the nematode? What is the benefits we are getting after studying the nematodes? And yes, during our in my previous slides, we had seen how the nematode, they are related to the sustainable goals. Okay, if we highlight this nematode, then we can realize that four sustainable goals, that is 2, 13, 14, and 15. Now, what are the other benefits of these nematodes? So first thing is that they are most abundant and multicellular animals, important for the crucial for soil health and nutrient cycling. Yes, our India is agricultural country, and these are the hidden enemies of agricultural field. These are the hidden enemies because as the plant, as the vertebrate or the animal nematodes, which you can able to see with our naked eyes, but particularly this plant nematode or agricultural nematodes, they are almost like the hidden. They are unable to see with our naked eyes. That's why the study of nematode is very important one in agricultural importance. So these are the model organism like seed eagles. There's 4 Nobel Prizes already there. Then the medical and veterinary significance. Yes, the medical importance. We know Ascaris lumbricoids, and we also see the how they do the damage. So there is one case here in Pune with the hospital. One lady was admitted because of the severe abdominal pain, and when the doctor went through the CT scan and all these things, they get surprised. They recovered near about 3,000 ascaris from that particular lady from the intestine, and the image was very disturbing. So I am not sharing that image, but totally intestine was very. So she was unable to even eat anything or drink anything. So the nematode, they are medically important and is veterinary importance, particularly sheep, goat, and the birds, poultry industry, okay, and allied agricultural fields. Veterinary importance, yes, lots of damage is done by this nematode into the veterinary field and medical. That's why the study of the nematode is very, very important. Yes, we can also explore this nematode for the environmental monitoring to see the pollution, impact of pollution onto the different type of habitat, and we can also use this as the climate research. Yes, and biotechnological and pharmaceutical potential is also there because lots of we are working on the nematode proteins. So in last slide, I'm going to show that how in my lab on what aspects we are working. So nematode proteins we can explore for the pharmaceutical potential also. So these are some data, statistical data. So these are the 57 billion nematode is there for every human. If you compare the population of the human with the nematodes, you can see for every human there is a 57 billion nematodes. So you can just multiply 100 and just India's population 140 million humans, and for one human there is 57 billion nematodes. So you can't imagine. That's why the biomass of the nematode is also heavy. So they are most abundant animal group on the earth. Around 80% of animals are nematodes. Okay, so this very interesting one as the insect and these nematodes. Both these groups that occupy heavily. So these are the ones critically very important for the carbon and nutrient cycling and influence the carbon dioxide emission. So in the sustainable development, we need to consider the increasing about the CO2 emissions. How this nematode we can utilize to reduce the CO2 emission, we can also think about that. And they're estimated between 1 lakh to 10 lakh species. So again, these are the described and described species. The most abundant metazone on Earth, crucial for soil health. Yes, so basically, if we see the classification in our previous slides, we had classified, but there are three to four different levels of classification is there. We can classify the first major three groups is there. One is a free living nematode, another one is a parasitic. You can see here the plant parasitic or the animal parasitic, and these are the two major groups for the classification of the nematode. Okay, and this one is the with plasmids and without plasmids. So these are, we need to consider these two characters for the classification of the nematodes. And yes, these are the different applications, how these nematode, particularly in case of the plant, they damage the plant cell, how they are responsible for the decrease in as the crop yield going decreasing, and these predatory nematodes, how they are predatory nematode, they are important for controlling these soil dwelling insect larvae. So then these are particularly in case of the human being, these are little human being like the hookworm. These are the Ascaris lumbricoids. These are the C. elegans. So these are very important model organism, C. elegans. And the aquatic habitat also, and these are the plant parasitic and human animal parasitic. So these are the levels of classification. So these are the levels of classification. So here, these are some typical images which we had already learned in our textbooks. How very interestingly, when we study nematodes, we concentrate on the adult one. But in most of the nematode cases, the adults they are not pathogenic. Very few cases adults they are pathogenic. Mostly they are larval stages. These larva, first larval stage, second larval stage, third larval stage, and up to these stages, these three stages, larval stages, they are very much pathogenic than adult. But while controlling the nematode population, we target most on the nematodes. And another thing is that on a very serious record, I'm talking on this. While identification also, we mostly reliable on the adult stages, but only adults particularly in case of the nematology, only adults they are not helpful for the identification. Yes, we use the adult stages, but we need to consider the life cycle of that particular nematode. Then only we can come to a at the egg stage, it is difficult in this nematode, that is but the nematodes, these are kinds of the damages. These are some particular. So just see the third one, EPN. At one side, this first free living and plant parasite is a harmful one. Habitat. Yeah, to buy the same habitat. That is their plant parasitic. Yes, no doubt they are present in the. That's why into the integrated approach of the nematology, we need to to uh, we need to kill, or we can say we need to manage this plant parasite, and there is need to enhance increase the population of EPN and the nematodes. So very interesting. That's why nematode
ढोले सर काय झालं हो? हॅलो हॅलो हा हॅलो
हा सर
ते कॉल करायला मी का टेक्निकल
सर